Thanks everyone for joining today. I'm happy to be here and kicking off the first webinar of our lichen project. It is exciting to share some results from one study of this project from a 24-year trial. So first, I want to acknowledge that Ken Rod and his team designed the experiment and set up the trial in 2000. Susan Kale and her team collected field data in 2016. We thank ARCKP and NSERC for funding this study. I would like to acknowledge ARCKP for their continued support and advice throughout the tenure of this project. We also thank West Fraser for providing study sites, providing the funding and data collected in 2016. Special thanks to Portage College for providing us with an excellent professional biologist to join this project. So in this presentation, we will begin with a bit of background. The first part will include project background, introduction of lichen, terrestrial lichen community, and terrestrial lichen restoration. And in the second part, we will move on to the research objectives, methodologies, and some results and conclusions from this study. So this study is just one of the big projects. And in this project, our goal was to evaluate feasibility of a terrestrial lichen transplantation in forest harvest blocks. So it's funded by ARCKP from 2021 to 2023 to find a solution that balance forest activities and the product of caribou. And then we received the NSERC funding to continue this research from 2023 to 2025. So we began with the review of current research and techniques, and we contacted some researchers who transplant lichens before in some sites and revisit three historical sites. One was in West Central Alberta. It is the tree harvest sites, which I'm going to present some results from these sites in this webinar. And the other two are the post-fire trails in BC. We also conduct a greenhouse trail and set up field trials to examine the effect of lichen fragment size and substrate on the successful of lichen transplantation. Additional, we also examined the effectiveness of hydroseeding for transplanting lichen. So we are going to have a series of webinars to share these results. Now we move on to the introduction of lichen. So lichen consists of a symbiotic association of algae, cyanobacteria, and fungi. The fungi provide structure and a source of inorganic nutrients for photosynthesis. Algae or cyanobacteria provide carbohydrate through photosynthesis, and the cyanobacteria can also fix and provide nitrogen for the fungi. So the component body of a lichen is called cellulose, as this picture showed. So the body is anchored to its substrate by the hair-like growth called rhizome. There are four main lichen body growth forms: crustacea, squamulose, foliums, and fructose. So both crustacea and squamulose are tightly bound to their substrate. The Kratos lichen forms a crust or coating on a substrate, but a subcumulose lichen consists of leaf-like scales. Foliar lichen is also leaf-like, but it has the upper and lower surface. The last one is the fructose lichen. It can be hanging or upright and maybe hair-like, cup-like, or shrubby, like this picture showed in appearance. Lichens are found worldwide and occurred in a variety of environmental conditions. Lichens growing on the ground are known as terrestrial lichen. Terrestrial lichen may have a higher abundance than a boreal lichen, which grow on trees. The majority of lichens are desiccation tolerant, so they can survive dry out and grow in some habitats such as dry and sandy sites, older and open peatlands with a dry surface layer. Terrestrial lichen communities are dominated by species like Cladonia, like the reindeer lichen showed in this picture, sterile column, like snow lichen here, Peltigero, like dog lichen here, and the last one is a centuria, like island moss, but it's a lichen. Lichen communities are important in the forest ecosystem. Woodland caribou often rely on Cladonia subgenus Cladinia as a primary food source during winter. Lichen usually account for 60 to 83% of winter diet for caribou. Lichen are long-lived. For example, Cladonia stellaris can live up to 120 years. However, lichens are extremely slow-growing, with a linear growth rate around 5 milliliter per year. And they are also poor dispersers. So there are some questions about how they propagate. Actually, lichen can undergo asexual and or sexual reproduction. Like a Cledina reproduce asexually through fragmentation and is dispersed through wind, rain, and wildlife. So during dry periods, lichen readily desiccates, but this also makes it susceptible to fire. So the increasing fire plus human disturbance result in rapid habit change and decline in resource. The research suggests that after fire, it takes 50 to 70 years of forage lichens to reestablish in suitable stance. Now we move on to the terrestrial lichen restoration. Techniques for restoring terrestrial lichens focus on transplanting or fragmenting lichen silage. So a lot of questions come up about health and long-term survival of transplanted lichens. But it is difficult to compare the success of different methods across different studies. In this study, we aim to evaluate the success of lichen transplants within forest harvest blocks. So the objectives was to determine if lichen transplantation result in increased lichen cover, compare lichen covers with different transplant techniques and lichen species, examine the relationship between success and cover of non-target lichens, mosses, forbs, woody plants, and graminoids. Examine the effect of key habitat variable. I will see set characteristics in the following slides, which include crown closure, tree height, slope, surface shape, and the percentage of surface substrates. and evaluate lichen dispersal beyond the experimental units. And here I have two pictures here. It shows the site in 2000 and 2024. So we can see a lot of change in the environmental conditions in this site from 2000. So the study area was located within the Berlin 1 and 21 compartments of West Fraser of Canada Hinton Division's forest management area. It's located between Fox Creek and White Cloud. Original forest in this study area include logical pine, and the area has been previously documented as woodland caribou habitat. Berlin 1 was harvested in 1994, and Berlin 21 was harvested in 1998. Lichen re-establishment trials was set up in 2000. So 7 transect arrays were established within 7 separate harvest blocks in Berlin 1, and 3 transect arrays were located within a single large harvest block in Berlin 21. This picture showed one transect array at a study site. So each transect array included 3 transects established at equal distance and directions from each other. Transects include control, hand transplant, and broadcast transplant at equal site at 0, 120, and 240 respectively from the array center. So the hand transplant plot to try to minimize the overall stress to the lichen clumps from physical transplantation. And the broad transplant was to emulate broadcast transplanting of lichen fragments at an operational level, such as dispersal by ATV or helicopter. And in each transect, it includes 5 50 centimeter by 50 centimeter plot. And the first plot was set up at 20 meters away from the array center. And in each plot, it includes 3 lichen species, C. mitis, C. rangiferina, and C. anciliars. And this picture showed how they set up the experimental plot in one transect. So lichen covers were assessed in 2016 and 2024. In 2016, Susan's team also collected the data of percent coverage of non-target lichens and general plant groups. And in 2024, we collected the data, the ecological data, including crop closure, tree height, slope, surface shape, and surface substrates. And in the same year, we also assessed lichen dispersal beyond experimental plots using a sequence of plots perpendicular to the center plots on either side of each transect. So this picture just shows how we designed. We flipped the quadra 10 times in each direction. So we measured 5 meters from the center plots. This slide shows the lichen cover in 2000. So King and his team collected this data. Before transplanting, the mean cover of C. mitis ranged from 0 to 0.6%. C. rangiferina and C. anciliars showed mean covers below 0.1%. After transplanting, each of the 3 species provide about 10% lichen cover in hand transplant plot, so totally around 30%. And the same amount of lichen we used in broadcast transplant plots, but was crushed into fragments, providing about 80% cover of mixed lichen. And this one just shows a empty experiment plot in control. These three pictures showed one plot from each treatment in 2016, and these three plots show one plot from each treatment in 2024. So in 2016 and 2024, blue sticks marking the array centers were found in most cases, and most treatment plots were located in these two revisit times. It's not hard to find them because sometimes you can use the dispersal lichen there as marker even without the flag. But it's hard if there's no flags to find the control plot. So several control plots were not found, and the new control plots were marked in 2016. And then in 2024, we did not find two transect of the control plots. So the new one marked and measured in the same approximate locations. So we combined the data on lichen cover collected in 2016 and 2024 and used the generalized linear mixed effect model to assess the difference in lichen cover amount treatment, lichen species, year, and their interaction. So the result reveals that the effect of treatment, year, the interaction between treatment and the species, the three-way interaction among treatment species and year was significant. So I'm going to plot out the mean lichen cover based on the three-way interaction. This slide includes a lot of information. I'm going to break it down into 4 points. So first, the cover of 3 species together within broadcast transplant plot and hand transplant plot were close to 30%, which was the original cover in hand transplant plot. It appears to be dramatically lower than 80%, the original cover in broadcast transplant plot. So it may be that some of the original lichens in the broadcast transplant plot died, perhaps immediately after planting, before the lichen was established. So second, both transplant plots had significantly higher lichen cover than the control plot in 3 lichen species across both years. So we can see that here. So it indicates that the transplantation of lichen significantly increased lichen cover compared to the control plots. Third, there was no significant difference in lichen cover among 3 species within each transplant transect in 2016 and or 2024. So it indicates that transplanted lichen survived regardless of lichen species or transplanting techniques. So last one, I want to mention that the thematics had a significant higher cover than the other 2 species in the control plot in 2024. So this picture shows what it look like in one control plot. For the data from 2016, we include the cover of non-target lichens and other plant groups as covariants in the model to understand their effect on differences in lichen cover. The results showed that treatment had a significant effect on the cover of all three species, where the cover of moss and graminoids had no significant effect on the cover of any species. The effect of non-target lichen and FOB cover was significant for the cover of thematics. The cover of FOB and woody plants had a significant effect on the cover of say ancillars. So I'm going to plot out the relationship that I marked here. So the relationship between the cover of thematics and non-target lichen was weak but positive. So as the cover of non-target lichen increased, the cover of thematics also tend to increase slightly. However, we have no observation of a high thematics cover with a high non-target lichen cover here, which result in greater variability in prediction at this higher value. So this result indicated that the competition from non-target lichen may not adversely affect the growth of thematics. The relationship between the cover of thematics and FOBs was negative. As the cover of FOBs increased, the cover of thematics tend to decrease slightly. So a positive relationship between say ancillars and FOB cover was observed. Similarly, there was more uncertainty at higher FOB covers level due to the limited number of high say ancillars cover with high FOBs cover in our dataset. The other graph showed a negative relationship between say ancillars cover and wood plant cover, which suggested that say ancillars may prefer more open area compared to densely wooded area. For the data from 2024, we also analyzed the differences in lichen cover among treatment and set characteristics. Same treatment had a significant effect on the cover of all three species. The effect of slope was significant for the cover of thematics and say ancillars, while other set characteristics showed no significant effect on the cover of any species. In these two scatter plots, most observations are clustered at low slope, with very few instances of high cover of thematics or the ancillars on steep slopes. A negative relationship was observed between slope gradient and the cover of these two lichen species. They tend to prefer flatter terrain over steeper slopes. Flatter terrain may provide environmental conditions such as increased soil moisture that are more suitable for the growth of these lichen species. In our dispersal trials, we analyzed the differences in lichen cover based on dispersal distance lichen species and their interaction. The species had a significant effect on lichen cover for all transects. Both the distance and the interaction between distance and the species had a significant effect on lichen cover in the transplant transect, but not in control transect. In the control transect, thematics had a significantly higher cover than the Rangiferina and the ancillars, and it had the highest variability. Distance did not strongly influence dispersal in the control transect, as most value remained very low. But we can see some high value for cymatics at greater distance, suggest the cymatics may come out on its own. And this picture show some quadra plot had a high abundance of cymatics, which results in one of these dots here. In both hand transplant and broadcast transplant transects, most data points remained at low cover values, but there are a few extreme outliers, particularly for cymatics in hand transplant transects. But in broadcast transplant transects, all the lichen cover lower than 6%. In both transects, all three species showed a general trend where lichen cover decrease as distance increase. This suggested that lichen dispersal at a short distance from the source. Cymatics had the highest cover and the broadest confidence interval at the pink color shown here. So this indicates that more variability in dispersal success of the cymatics. C. rangiferina and C. ancilius had a lower cover, with C. ancilius showing the latest dispersal success. Now we move on to the conclusion. Transplanted lichens survive well over multiple decades and increased lichen cover compared to controls. So this result indicates that lichen transplants can be utilized to restore caribou habitat impacted by tree harvest. So that means lichen can be survival after transplant, but just grow slowly. In terms of the methods, broadcasting fragments versus planting clumps produce the similar covers. This means that we can use machine to disperse lichen fragments over larger areas in short time frames. So it will be more economical compared to hand transplant methods. Lichen covers were better on lower slope, but were not associated with any other measured site characteristics. I want to mention that there was no significant relationship between pine needle substrate and lichen cover, but we still observed that plots had high pine density usually had little to no lichen present. Cymatics cover was positively correlated with the cover of other lichens, but negatively correlated with bulb cover. The cover of C. ancilius was positively associated with bulb cover, but negatively correlated with wooden plant cover. But there was a considerable variation in success with both treatment. Some plots contain no lichens and other contain much more lichens than the original covers. So this variation was observed both within and between transects. It indicates that microsite characteristics beyond what we measured right now may impact the success of lichen transplants. Lichens did not dispersal significantly beyond 50 centimeter, especially for C. rangiferina and C. ancilius. But there is a limitation here. We only use a small quadra to measure two direction, but not a circle with a long distance. So it's only capture limited materials for dispersal. Overall, this historical sites provide a lot of valuable information for us. to examine the feasibility of lichen transplantation for the caribou restoration. Thanks for your attention.